The workflow of the Make It system starts with a fluid handling robot that is used for automated preparation of samples and also transferring of these samples from 384 well plates to a DESI plate using a pin tool. The DESI plate is just a glass slide with the footprint of a well plate that is coated with a porous PTFE membrane. We've demonstrated the use of this system for different sorts of samples, from organic reactions to enzymatic assays to straight up biofluids or bacteria. Once done, the plate is put on a shuttle, sent to the other side where a robotic arm takes it and places it on the DESI stage. Before the analysis starts, the only thing that we need to do is calibrate the positions of 3 marks on the corners of the plate that will allow us to triangulate the positions of all the other spots deposited. We can put up to 6,144 spots on a single DESI plate, and this is done by offsetting the pin tool a little bit in between each pin. We have 16 possible different offset, and the software allows us to select how many of those we pin. We don't have to pin all of them at the same time. In this case, for the demo, I pin just 4, which represents 1,536 samples. The analysis of this number of spots takes about 25 minutes. The software also provides great flexibility because you can design any instrumental method in any way that you want. You can select, for example, the tune file that you want, the mass range that you're interested in, and you can select any scan type that the instrument allows you. So you can perform any mass spectrometer experiment that the instrument accommodates, just in a high throughput fashion. Once it's done, you save all this and you start the analysis. So here we're going to see the live acquisition of spectral data from the plate. This counter indicates how many spots have been analyzed, how many are left, and so the analysis will start now. This particular plate contains enzymatic assays, in this case of acetylcholinesterase, which is an enzyme that catalyzes the hydrolysis of acetylcholine 146 to choline 104, and those are the peaks that we're seeing right now on the screen. As you see, all these different flashes corresponds to the spectral information from each one of the spots. Some of the spots are empty, that's when you see that the signal goes down. But the intensity ratio is what we're going to use to then calculate the progression of the reaction, the inhibition, if we have a particular inhibitor in a mixture, and so on. This analysis is carried out straight from the bioacid mixture, which in this case is PVS, which is a buffer containing phosphates, containing NaCl. It also contains BSA, the enzyme, the substrate, the product. So it's a very complex mixture that is analyzed directly with no workup. This is the stage moving in real time, jumping spot to spot instead of rastering across, which increases the speed at which we can acquire spectral data. Once the analysis is complete, we end up with a single raw file which contains all the spectral information from the plate. As you see, if we move across, we're going to see the spectrum corresponding to each one of the different spots. And so we can automatically process all this spectral data. The only thing that we need to input is a plate layout file, which looks something like this, which is a table containing the M/C values that you're expecting. To this table, you can add as many rows and columns as you want. That's particularly useful for organic reactions, cases where you're screening different combinations of reactants A's and B's, and the intersections you can put the masses of the products that are expected. In this case, I look for a single product in the whole plate, so I just have this cell. Then you can put the layout of the reactant A's and the reactant B's. This case is simple because I have only one combination that I'm looking for, but you can put as many combinations and in the many different places that you put within the plate. You can add as many worksheets that are going to work as descriptors of the data. In this case, I just have indexes, but you can, for example, put here ACN, methanol, DMSO, if you were doing organic reactions in different solvents, or here catalyst and not catalyst, depending on whatever conditions you're interested to define your data with. And this last worksheet just contains, again, a selection of which one of those 16 possible offsets did you actually pin on. Once this is done, you can just select the plate layout, the folder containing the data, which in this case is demo, and then we start the data processing. The software will automatically convert the raw file into a text file and start parsing through it, assigning all the scans to different spots, calculating the average spectrum associated to each spot, and then parsing through that information, finding the intensities, the maximum and average intensity of the ions that I was interested in this case 104 for choline and 146 for acetylcholine. Once it's done, the results can be easily visualized in an interactive heat map like the one shown on the screen. The heat map can be filtered by the M/Z values you were interested in. You can click any particular spot and see the spectrum associated to that spot. You can also filter by intensity or any of the descriptors that you added to the plate layout file. All this information is also exported to a CSV file, and you can use it to process further, for example, to create those response curves.